Hi, thank you for that kind introduction. For the next topic, we'll be discussing drug substance facilities, in particular hidden facilities and critical intermediate facilities used in the manufacturing of drug substances. Again, my name is Cassandra Abellard. I'm a consumer safety officer from the Office of Pharmaceutical Manufacturing Assessment. My colleague Wei from the Office of New Drug Products will give the second half of this presentation, which focuses on critical intermediates. As a reminder, please type any questions in the Q&A box so we can answer these during the live Q&A following the presentations. So when it comes to drug substance facilities, often there's some confusion or lack of clarity on which facilities involved in manufacturing need to be listed in applications. During this presentation, we'll attempt to clarify which facilities should be listed. The following facilities in the DMF are recommended to be included on the NDA and/or ANDA 356H form. All final drug substance manufacturing, micronization, sterilization, and testing sites that are produced or proposed to be involved in the disposition of commercial product. Sites for intermediates addressed by ICH Q7. Facilities used for storing or warehousing drug substances prior to disposition decision, including those that solely store the stability samples. The required facilities that are in the DMF but do not appear on the NDA and 356H form are referred to as hidden facilities. For an ANDA amendment involving facilities, any amendment that includes a new facility that requires comprehensive evaluation is considered major. So in the DMF, these situations may arise, and they include the inappropriate selection of a regulatory starting material. If there is a reference to a secondary DMF whose product may be a critical intermediate, any outsourced API test sites, or any outsourced intermediate manufacturing sites. For example, maybe a micronization site or a sterilization site. As most of you are aware, ICH Q7 represents the agency's current thinking on GMPs for APIs. Section 501A2B of the Act requires that all drugs be manufactured in conformance with CGMPs. This includes APIs. API and related manufacturing and testing facilities that follow this guidance generally will be considered to comply with the statutory GMP requirement. Again, as most of you have previously seen, this is a section of the table located in ICH Q7, which outlines the increasing GMP requirements depending on the stage of manufacture of the drug substance. To note, an accepted regulatory starting material is the starting point of GMP. Now, accepted means accepted by the agency. And the manufacturing process of all intermediates is subject to GMP requirements. To give you a brief overview of the current hidden facility identification process performed during the DMF assessment, there's two stages. The first is the TCIR. TCIR stands for Timely Consults and Information Requests. During this process, which is performed during all original ANDA applications and resubmissions under GDUFA 2, we're looking for facility discrepancies between what the applicant lists on their 356H form for the API sites in the ANDA and what the DMF lists for these sites in the DMF. So comparison is done between the application and the DMF. We will then issue comments to the applicant when there are discrepancies or ambiguities in the facility information. And during this process, we also evaluate if the regulatory starting material is acceptable for original DMFs, and we'll issue comments to the DMF holder as necessary. We also perform a DMF CMC assessment for all NDA and ANDA applications. During this, we identify DMF facilities which are required to be listed on the applicant's 356H form based on facility functions. We check if required facilities are listed on the applicant's 356H form, and we issue comments to the DMF holder and/or applicant for any discrepancies or ambiguities. So that comment that may get issued, as I keep referring to, we'll issue a comment. This is what it may look like. I'm not going to go over this entire comment. However, I wanted to give you the full text as to what you may see. It basically tells the applicant to contact the DMF holder and update their application. The text does get modified depending on the situation. However, I did want to note the two bolded sentences. I will go into further details around those on the next couple of slides. So the first bolded sentence that I wanted to highlight and try to explain in a little more detail is kind of in reference to a potential secondary DMF that is maybe involved in the application and maybe is not listed, which would then be considered a hidden facility. So basically it says there's a facility that's included in the DMF, a DMF that is referenced by a primary DMF that was not listed in your application. Please note the agency cannot provide you the status of facilities not listed in your application. So some key points around that. The LOA that's included in the application makes the DMF part of the referencing application, and this includes the facilities in the DMF. But when a secondary DMF is referenced, it includes those facilities in the secondary DMFs as well. So a little pictogram of what this is trying to say shows you the final drug substance G on the right hand side. That's typically the DMF that is referenced in application. However, the manufacturing of this DMF may reference other DMFs, which may also contain critical sites that require evaluation. If these secondary or sometimes even tertiary sites are not listed, we can't share the facility information with the applicant. So the second bolded sentence of the hidden facility comment that I wanted to discuss comes from this sentence, and the bolded part is what I wanted to go over. If only a subset of the facilities in the DMF will be referenced by your application to support commercial manufacturing and/or testing, your LOA should specify those facilities. So often there are multiple intermediate sites or perhaps a micronization site that are listed in the DMF. However, the applicant is not using all the intermediate sites, or maybe the drug substance is not being micronized for their product. Based on that, it's important to list all required facilities in the 356H form. This will facilitate the application assessment process, the communication on application status between the agency and the applicant, and if only a subset of facilities in the DMF will be referenced by the drug product application, those should really be specified in the LOA. Whether or not a DMF facility is listed in the application will not change the determination of whether a facility needs comprehensive evaluation, and therefore may cause potential barrier for application approval. For example, if issues are noted with a hidden facility that's not listed in the application, and the agency has determined these sites are critical and require evaluation, we will still review those. facilities. So as a summary of the hidden facilities we've seen in the past several years, as you can see from October 2017 to about December 2020, the total number of unique ANDAs evaluated was about 2,700, and the number of applications with at least one hidden facility being noted was over 400. And the total number of hidden facilities identified was 553, which means that several had more than one hidden facility. So the overall percentage of ANDAs with a hidden facility issue or concern that had to be addressed was about 15%. Specifically, the types of hidden facilities can be broken down as 31% were manufacturing facilities that were hidden or missing from an application, 45% were testing facilities, and about 24% were critical intermediate facilities that were not listed. This high number is indicative of poor communication between the applicants and the DMF holders regarding the DMF-related facilities that support their ANDAs. So how can industry improve? List all required facilities in S.2.1 in the DMF. The final API manufacturing and testing sites, intermediate sites performing operations addressed by ICHQ7, facilities used for storing or warehousing the API prior to disposition decision, including those that solely store the stability samples. Clearly indicate the responsibilities of each facility in the DMF. If it's an intermediate site, indicate which intermediate is being manufactured. If it's a testing site, indicate which tests are being performed. And communicate all required facilities and their responsibilities early to the NDA and/or ANDA applicants. The agency considers all facilities listed in a DMF applicable to the referenced application, unless explicitly stated in the DMF LOA that only certain facilities will be used. Again, required facilities in the DMF include final API manufacturing and testing sites, including any alternate sites that may be used. This includes API manufacturers, API routine commercial release stability testing sites, including micro-testing, facilities that micronize the API, facilities that directly sterilize the API, and for animal or plant-derived products, the facilities that perform crude extraction prior to purification of the API. And now I'll turn it over to my colleague Wei, who will further discuss critical intermediate sites.
Thanks. Hello everyone. This is Wei Liu from Division of Lifecycle API. Now we are going to discuss how to evaluate whether an intermediate in your manufacturing process is critical or not. Critical intermediate is an intermediate whose manufacturing process is considered so important to the quality of final drug substance. So its manufacturer site needs to be part of the facility evaluation for the drug product application. Determination on critical intermediate is made on a case-by-case basis using principles in ICHQ 11, as well as the supporting data in submissions. During the DMF assessment, the DMF assessor makes a recommendation based on the manufacturing process of the drug substance. The Office of Pharmaceutical Manufacturer Assessment makes the determination if an intermediate facility needs a comprehensive evaluation. This slide shows how to evaluate whether an intermediate is critical or not. Assume we have a manufacturing process like this. It starts from the starting material A and B. After several steps, it produces the final drug substance G. Here, A and B represent the acceptable regulatory starting material, which complies with ICHQ 11. The CT here means chemical transformation. You may refer to ICHQ 11 glossary for this concept. There is a clear relationship between the risk and the number of steps from the intermediate to the final drug substance. As the process moves forward from A to G, the criticality of intermediate increases. Specifically, we will consider the following factors when we determine whether it is a critical intermediate. First, overall synthetic scheme and the complexity of chemistry and the final drug substance. We will consider how many steps away from this intermediate to the final drug substance. Whether the drug substance quality can be mitigated through the intermediate specification. If there are no adequate acylation steps from the intermediate to the final drug substance, or the drug substance is very complex, this intermediate introduces the most critical structure features to the drug substance. The intermediate will be considered critical. We will also look at whether the intermediate process is using common chemistry and reaction conditions, or using complex sensitive chemistry. If the submission includes the supporting data to demonstrate the impurity carryover has been adequately controlled, the risk will be lower. Otherwise, it is higher. Additional consideration is in-process controls and the intermediate specification. For example, with the same acylation steps from the intermediate to the final drug substance, an intermediate with purity of 85% and an intermediate with purity of 95% will bring different levels of risk to the drug substance quality. In most cases, in situ intermediate doesn't have opportunities to remove impurities because it doesn't have the acylation steps. So we don't consider it as a separate chemical transformation step. At the same time, we also understand in some rare cases, an intermediate is unstable and very active. The chemical reaction is very straightforward. In this case, it's okay not to acylate the intermediate, but please include your justification in the submission. The determination of critical intermediate is based on complete manufacturing process from the regulatory starting material to final drug substance. When the whole process is covered in one DMF, the regulatory starting material is the materials listed in section 2.3 in the DMF, and the complete manufacturing process is described in section 2.2. When a DMF references a secondary DMF, the acceptable regulatory starting material may not be the material listed in section 2.3 in the primary DMF. Let's continue to use previous case as an example. We assume the secondary DMF covers the manufacturing process from A and B to D, while primary DMF covers the process from intermediate D to final drug substance G. Now D is a product in the secondary DMF. It is also listed as starting material in section 2.3 in the primary DMF, but is not the regulatory starting material in this process when we consider the complete manufacturing process from A to G. It's actually an intermediate. Whether intermediate D is critical or not depends on the risk assessment for the complete manufacturing process from A to G. That means we will evaluate the complete process included in both primary DMF and secondary DMF to make the decision. As we mentioned before, we will consider the complexity of the drug substance, the number of steps from intermediate D to final drug substance G, in-process controls, and the intermediate specifications. The inclusion of secondary DMF can be extended to a chain of secondary DMFs. For the process from A and B to G, we assume the primary DMF number 1 covers the manufacturing process from drug substance base F to final drug substance G. It references the secondary DMF number 2, which covers the process from intermediate D to F. The DMF number 2 references another DMF number 3, and the DMF number 3 covers the process from the acceptable regulatory starting material A and B to intermediate D. In this case, the drug substance base F is generally considered critical, and its manufacture facility in DMF number 2 will be evaluated. For intermediate D, we still use the principles we just discussed to determine whether it is critical or not. If it is considered critical, its manufacture facility in DMF number 3 will be subjected to evaluation. Here I would like to clarify, the complete manufacture process from A to G is included in one DMF, or separated into several DMFs as primary and secondary, would not affect the decision whether intermediate D is critical or not. Another point I want to mention is, the same intermediate which is non-critical in a process may be considered critical for a different manufacturing process, because we have more than one factors needs to be considered. For example, erythromycin A is derived from fermentation process. It is used to manufacture drug substance clarithromycin and erythromycin. Whether this fermentation product is critical or not would be determined by its own specification and its subsequent process controls till final drug substance. It could be considered non-critical for the drug substance of erythromycin, but critical for the drug substance of clarithromycin. Same principles also apply to post-approval changes. After the DMF becomes adequate, if you want to add a new manufacturer for the intermediate, please provide the following information in the submission: name, address, fee number and DUNS number for the new manufacturer. An evaluation of potential impact on impurity profile of intermediate and final drug substance. Again, we consider the same principles to determine if this intermediate is critical or not. Another point I want to mention is, DMFs intended for reference in a generic drug submissions that are subject to the DMF fee under GDUFA may only contain a single drug substance process. So if you would like to qualify a different vendor for the intermediate, please keep this in your consideration. This slide summarized this presentation. In Part 1, DMF hidden facility identification has been discussed. It is a potential barrier for application approval. In your DMF submission, please list all required facilities and their responsibilities in Section 2.1. Please communicate early with drug product applicant to facilitate the application assessment and application approval. Then we introduced how to evaluate the critical intermediate. Determination on critical intermediate is made on case-by-case basis with risk assessment. Appropriate control strategy with supporting data may help to lower the risk. This is our acknowledgement. Thank you. If you have any questions, please type them into Q&A box in the bottom right-hand corner of your screen so that we can address them during the Q&A panel after this session. If you have a question on this presentation after this workshop is over, please send them to the email address provided here by March 19th for inclusion in the follow-up webinar. You may also refer to the following presentation for additional information. Thank you for your attention.